Hi, everybody. Can you hear me? Excellent. So I'm putting this watch down here so I don't overstep my limits. Thank you very much for the invitation for this meeting. And I'm going to present you something which is absolutely basic research. It's about the de novo synthesis of life. De novo, that's a chemical term. It means that you do it from scratch. You do it from bottom up. And it's, in a way, it is actually a chemical project, as you will see. What I also want to emphasize on that on the top right, you see the ERC. There's the European Research Council who are supporting our project. So let's get down to business. These are the cardinal questions, right? Why are we doing this? What on earth are we doing? Who are doing it? How and when? And I'm going to address these questions in turn. So why are we doing this? Well, I think because we want to obtain a proof of principle, which means that you have an idea about something, how nature works in a certain domain, and then you want to prove that the idea is correct. That's the proof of principle. In fact, this is a very interesting thing for Hungarians because there is a Hungarian allusion here, which is very strong, right? That comes from this gentleman, Imre Madács, of course you will know. And there is a marvelous English translation. It's actually a little bit old English, so it fits the language of Madács extremely well. Why man too almost, if he should but learn, might in his kitchen cease as good a broth? In thy great kitchen thou hast placed man and seest indulgent how he spoils the food. A very bungler thinks himself a god. So why are we going down this line that, of course, is uttered by Lucifer, the devil? So what are we up to here, really? Well, this is exactly the proof of principle. And this is Richard Feynman, very important theoretical physicist. He wrote very famous books, also available in Hungarian, the Feynman Lectures on Physics. And he said, what I cannot create, I don't understand. And I fully agree with that. So unless we can create something which is a living system, we don't understand it. So what are we going to do? Certainly not this. This is a creature, created by Dr. Frankenstein. We don't want to create monsters, and that's one reason. The other reason is this is very complicated. You don't want to synthesize something like that de novo from chemistry. Now, these cells, our cells, the eukaryotic cells, are also far too complex, right? Not to mention the fact that they are inhabiting organelles that once upon a time were free-living, like the mitochondrion or the plastids, which means that they are higher up in the organizational hierarchy. Arguably, bacteria are the simplest autonomous living systems. Viruses are not autonomous. This is important. Yeah, but they look simple, but there is a problem, a very big problem. They all have a genetic code. The genetic code, the translation from DNA to protein molecules that do the actual work. Now, that is the result of a very long evolution. You cannot start with that, right? Okay, so it has to be something simpler. So let's get into this a little bit. What kind of things you have about life as leading ideas? One is, on the left-hand side, that there is some kind of a cyclicity to maintain your activity, to gain food and energy, right? That's on the left-hand side. It's a cyclic process. On the right-hand side, there is a program that is executed, right? From birth, actually, the death is not displayed there, but I can assure you it's going to await all of us. So it's a combination of two things, this cyclic process and this kind of a program-controlled process. And if you think about it, how can you achieve such things? That is by organization. Okay, how you combine the different parts? As you see, the bows and arrows on the right-hand side, this is the result, how the father was actually creating these things for the kid. So bacteria, just by looking at them, have three very important components. One is a boundary, right? And this was eminently discussed by our Nobel Prize winner yesterday. Then you have a huge metabolic network. It's a nightmare for the biochemistry students, but this is how it is. And this is the genetic material. These are absolutely indispensable components. Of course, you may not use exactly those in the same form because they are far too complicated. What I want to say that from the chemical point of view, and this is absolutely important, all these processes rest on autocatalysis. Catalysis is that something helps the formation of a substance, right? Autocatalyst is a substance that helps its own formation. So here is the rendering on the left-hand side. You have got these two little circles there, and it consumes two single circles as it turns around, and then you get two copies of the molecule. That is autocatalysis, right? And of course, it leads to exponential growth if the food is plenty, and then this is how ultimately biological reproduction and the growth of population is actually possible. But that rests on chemical autocatalysis, always. Let me tell you an example which we are also using when we make the constructions. So here is the foremost reaction. This is a real reaction. This is formaldehyde, this is glycolaldehyde. And there is this series of reactions, as you see, from glycolaldehyde of the left-hand side, it consumes two molecules of formaldehyde, and after one turn of the cycle, there are two molecules of glycolaldehyde. And this is the autocatalytic part, and that was discovered by Butlerov in 1861, much before the Miller experiments in the 1950s, by the way. And it doesn't need enzymes. Right? Okay, so DNA and replicability are very important, but again, this is very, very difficult to do. You know, in chemical terms, you need complicated enzymes, and this is exactly what we dare not use. So you have to go for something simpler. This is the famous case that was done by Günther von Kintrovsky in 1986. This is a self-replicating hexadeoxynucleotide, like a piece of DNA, but if you give these little pieces, which are trimers on the left-hand side, then they bind by Watson-Crick pairing, and if these molecules are activated, then the annealing, you know, by the Watson-Crick pairing helps the formation of the covalent bond between the two trimers. And because they are short, they can also spontaneously dissociate. And now you have two copies because they have palindromic sequences. So you take the copy, you turn it, and it's exactly the original one. So this is actually reproducing without enzymes. It's a very good lead. And there is the nature of the boundaries that was discussed yesterday. What I want to say is that in chemical terms, there is the process of spontaneous insertion, which can lead also to autocatalytic growth of the membrane surface. So all of these processes are autocatalytic. Now, who are doing this project? Here are the suspects, wanted, right? So there is one molecular biologist, Andrew Griffiths from Paris. There are two chemists, Gonen Ashkenazi from Israel, Sibren Otto from Groningen, the Netherlands, and yours truly. So this is the group, right? It's a 13 million project, 13 million euro project for 6 years, and we are just past the first 2 years. How are we doing it? Well, some abstraction and idealization is needed because, as we discussed, contemporary life is far too complicated, and it is a result of a very long evolution, right? 4 billion years. So something simpler has to come. Now it is expected in the emerging field of systems chemistry, the best guiding light actually comes from Hungary, and we should be proud of that. That's Tibor Ganti, who was a chemical engineer originally. And in 71, he published this landmark book, which was so clear that nobody understood it. Now, this is a paradox, but I mean it, actually. Why? Because everybody was fascinated by molecular biology at the time, and rightly so, but Ganti recognized that this is a no-go if you want to put the questions of the origin and the very simple synthesis of life. You cannot start with molecular biology. You have to do for something simpler. Here is the most recent edition in 2003. It contains actually several books now written by him and commentaries partly by me. And here is the scheme from 1974. There's a 74 scheme. This is a metabolic engine. It's an autocatalytic process, as you will see, because you see that's the part when you have the doubling. The guy is eating X, which is the food, is producing Y as a waste. Then this is the raw material for the genetic subsystem, and this is the raw material for the membrane. So this is autocatalytic, here is the doubling, and the membrane surface is also doubling here, right? Okay, and the system as a whole is also autocatalytic. And it can be proven because of geometric relationship that if everybody grows like this in a growing vesicle, then there will be spontaneous division because of physical-chemical forces. So that's the goal. And, you know, I mean, it's quite old now. It's about 50 years, right? Okay, that's another way of saying the same. I'm jumping over this. Now we are not there at the Trinity, the Holy Trinity in this case, when you have the membrane, the boundary, and some kind of a template together. That's the goal. What people are doing, including ourselves, is this, right here. Sorry, here, which I called infrabiological systems about 15 years ago, is the double systems, right? The third one is always missing. And this is the combinatorics that people want to realize in the lab these days, also including ourselves. This actually comes from 78, from Ganti himself. This is a self-reproducing spherule, which consists of the membrane and an autocatalytic metabolism, but no genetics is involved here. That's actually something which we have actually already realized. Here is it. So on the right-hand side, do you see the Formose reaction again? And this was put into little droplets, water droplets in an oil medium. And those droplets who have the glycolaldehyde inside, they grow, because of this autocatalytic growth. And then this is channeled through a system, a so-called microfluidic systems. So when the droplet becomes too big, it is split into two by the forces that it is forced through a certain microfluidic channel. So that was done in 2024. What we also would like to do is, of course, some kind of genetics, right? And this is actually a very important thing because by itself, the Formose reaction is always the same, right? There are fluctuations in concentration of whatever, but the fluctuations you cannot inherit, and that's obviously a problem, okay? So, what can you do? Well, you can try to do something like inheritance with different autocatalytic cycles. If the cycles are actually helping themselves, there is some interconversion between the cycles, but only very small. So in this case, to cut a long story short, you can actually prove that it is very easy to find what is called hysteresis. So if, for example, this is the replication rate of one of the cycles, so if you increase it, it will spontaneously switch their state when this particular reproductive system is going to have a high concentration. Now, if you are here and you go backwards, it's not the same route, right? And here you have a region of bistability, right? So both states can be stable, a bit like two DNA sequences can be stable in the same environment. Okay, that's one line. The other line, much more familiar to us, is that we generalize the von Kidrowski mechanism for replication. And this indeed has been done. So look at this here. Here on the right-hand side, this comes from Gonen Ashkenazi. This is not nucleic acid, these are peptides, 32 amino acids long, but they have a surface which acts as a template. So if you put these two together, like in the original thing, but that was nucleotides, right? And then you have a spontaneous replication again at the level of the peptides, and it's much more easy to handle actually than nucleic acids. So that's one of the components that we are using for what we want to do. Non-biological components may be key to success here. That's very important, right? So you have very important molecules in contemporary biology, but they are too complicated and they are always together with enzymes. Almost everything that happens is due to enzymes. So it's a special world, highly evolved. If you use non-biological components, you may be actually more successful in creating a de novo life just by pure chemistry. And this is an example from Sieber and Otto. This doesn't look like DNA at all, but it's worthwhile looking at it. So these are the pieces. Then they can be joined. These are peptides. This can be joined by the disulfide bond. And then there is a ring here, and there are spokes. These arms are hanging down. Now, because of the surface of the rings, they can actually spontaneously assemble into these towers. I used to annoy him that these are the Otto towers on the example of the Trump towers, and he doesn't like that. But this is actually, these are secondary bonds. There is no covalent bonding here. But still it grows, right, if the building blocks are there. And then if it gets too long, it breaks. And then it starts again. So again, it leads to exponential spontaneous replication. And we see also the trace of protometabolism. I mean, you need luck in science. You have to believe me. You can be very smart, very diligent, whatever. Without luck, you are not going to get very far. So the luck is here. It turned out that this, by itself, can catalyze an important transition. So it can catalyze the formation of its own building block. And there is another reaction that it can catalyze, the reaction between these molecules to give rise to this one. And that can lead to coacervate droplets. A coacervate droplet is a blob of organic material which doesn't have a membrane, but it's still fairly stable. It's like the membraneless organelles in contemporary biology. So now, if you look at it, then this is coming dangerously close already to what we want. Okay, you have the replicator, you have some kind of a protometabolism, and there is the formation of these blobs, the coacervates. Okay, that's just another rendering of the same idea. I will jump over it, but this is actually a concrete vision of the realization of the tripartite system. When? Now we are doing it because we are in the second year of the project. And you might ask, okay, are we going to succeed? Actually, I believe that we will be succeeding. We have 4 more years left, and we might actually succeed before that. I want to tell you also that this would have been impossible, completely impossible 10 or 15 years ago, because chemistry has developed tremendously in the meantime. So now you have knowledge, you have experiments, you have techniques techniques that were not available at the level of proficiency. So I think that's we are just exactly at the right time. Fortunately, the people in the ERC committee actually thought the same. So it's happening now, and let's hope that we are going to be successful with this endeavor. Well, thank you for your attention. That's actually the chemoton symbol by Ganti. It was designed by himself. You know, the 3 autocatalytic processes interwoven so that you form a unity. Thank you very much for your attention.
Thank you very much for the very interesting presentation, and I would like to ask the audience if there are any questions. Yes, please.
Is it on? It's on now. So I know that you actually started the presentation saying that it is very basic research, but do you think that this could have some clinical significance or implications in the distant or not so distant future?
That's a very good question, but it reminds me of, I think it was Benjamin Franklin, you know, dealing with lightings, electricity, and somebody asked him, okay, what will be the use of these games that you are playing? And then he said, well, I don't know yet, but I am sure that one day it will be taxed, right? And the electricity is taxed very nicely. So setting the jokes aside, I think if this is successful, what you can achieve, that you can actually produce custom-made artificial living systems that can do certain jobs that otherwise you couldn't do. And maybe not in the clinic, I don't know. Look, I mean, I'm a basic researcher, but it could be that in biotechnology, in the colonization of other planets, but I don't know. There can be many things, but I want to say that this is not foreseeable usually. So this is what the the politicians who want to support research don't understand. Okay, you should not ask immediately, okay, what is going to be the use of it, right? I mean, general relativity, which was created in 1916, was a marvelous thing, but people thought, okay, so what? Now it turns out to be that if you did not have the corrections of the Newton equations that come from general relativity, the GPS would be in error of about 200 or 300 meters, right? So this is the lesson that I'm always telling to politicians, okay?
Thank you very much. And there is a question there.
So thank you for this really inspiring talk. You told us lots of things about what's the theory behind this whole thing, but can you tell us a bit about how you are doing it? So what are the experiments? What you are doing?
Well, I mean, I showed several of the experiments, right? The compartmentalized Formos system was one of them, right? The Sebring, the Otto towers is a second one, right? A third component is the peptide replicators. What you have to do is that you have to put them together into the same system, and you have to ensure the coupling, right? And all this without enzymes, right? So you have to be tricky because that, of course, there is catalysis here because all the components are autocatalytic, it's fine, but it's very different from the catalysis that enzymes can do. So you have to locate the promising organizations in chemical space. And this is actually some of the things that we are doing. It's a good question because I can tell you. So we want to have more autocatalytic systems, also at the small molecular level, like the Formos reaction, right? So we use and develop something which is called generative chemistry. That's in silico chemistry. and there is a so-called graph grammar that you apply to the classes of chemical reactions, and then you can create chemistry in silico, and then you consult the experimental people, and then, you know, there is a give and take. So the experiments strengthen the theory, and the theory guides partly the experiments, okay? I hope I answered your question.
Thank you.
Thank you very much. And there is one more question from...
So I have a couple of introductory sentences before I ask my question. So many people think that COVID-19 was escaped from a laboratory. And nature wasn't ready for it because it wasn't created by nature. And my question would be, if you have anything put in your system which would stop these creatures if escapes from the control. And an example is that these days it's very fashionable to do cell therapy. In cell therapy, they put into the body cells which substitute for some function. But it's very important that what to do if these cells become cancerous. And to prevent that, they put a gene into these cells which is sensitive to a chemical which kills the cells if they escape from the control. So my question would be, if you think about putting something into this creature which can stop it multiplying, and because it's nature is obviously not ready for it.
Very good question. It's a justified question. But I'm happy to report that at the moment, and I think that for the foreseeable decade or so, there is no real imminent danger because they are so fragile, right? You sneeze in the lab and they will fall apart, right? So they are not like bacteria or viruses. They are very fragile organizations, and they will be for some time. Yeah, but you have to prepare exactly as you say. We are preparing, so we are actually already working on the guidelines, you know, for the future, how to attend to these things so that you don't food into your, well, food. Okay, let's be polite. And so we are conscious about it, but the danger is not imminent, but it is foreseeable in the more distant future.
Thank you very much. And if there are no other questions from the field, then unfortunately our time is up and we would like to conclude this session. But first we would like to join you, Professor, to ask you to join us, and we would like to provide you with a certificate of appreciation.
One more thing, that
yes,
I'm going to be here also during the lunch break, so don't hesitate to attack me. Okay, you are very welcome.